Now, Bismillahirrahmanirrahim. In this video of chapter number 51, we are mainly going to discuss how the contrast borders and how a flat light is going to affect our transmission in the rods and corn cells, from the rods and corn cells to the ganglion cells. So, what are these cells? These are either rod cells or corn cells, and they are going onto this cell, which is a bipolar cell, which leads onto the ganglion cell. Now we also have these horizontal cells which are mainly present in the outer um, plexiform layer. Now when all the rod cells are activated, these are all the rod cells, and when like all of these rod cells are activated, what will happen that due to their activation, this rod cell, the central one, is going to excite the bipolar cell, but the lateral two ones, they are going to excite the horizontal cells. And when the horizontal cells are excited, in this situation, what will happen? This main rod cell is going to act in the bipolar cell and it will give a depolarizing effect. Whereas when the rod cell excites a horizontal cell and it excites it mainly inhibits the bipolar cell, the mechanism is hyperpolarization. So this is going to be hyperpolarization, whereas this is going to be depolarization. But depolarization in this level is hyperpolarized by these two cells or horizontal cells. As a result, there is null effect and there is no light that is mainly transmitted. So this is the mechanism that happens in the case when there is like a flat light is coming. So what will happen in such a situation? So there will be neutralization of the whole pathway. But what happens if there is like a contrast border? In the contrast border, what happens is that the central rod is activated, but either one of this rod or another of this rod is activated. You can say that the central portion is activated, but one of these lateral ones are activated. What will happen due to this? The central will be depolarizing to the bipolar cell, the lateral will be hyperpolarizing to the bipolar cell. The hyper and depolarization, they will not be able to cancel each other, but rather they will produce a stimulatory effect. When there is one depolarization and two hyperpolarization, that is a effect, it is known as neutralizing effect. But when there is one depolarization and one hyperpolarization, there will be amplification of the signal, and due to that amplification of a signal, there will be a signal that will be transmitted by electronic conduction to the ganglion cells, and from there an action potential will be generated via these contrast borders to which will be carried to the brain. So this mechanism of lateral inhibition, it actually functions to provide contrast detection and enhancement. Now we are going to talk about how the colors are transmitted by the ganglion cells. A single ganglion cell, it may be stimulated by several, or it may be stimulated by only a few cone cells. This mechanism that we studied above, this was for the light and like for the black and white vision, like how we perceive white light when there's flat light, and how we discover the black by contrast border thing. That was it. Now we are going to discuss the color signals transmission, the color signal detection by the ganglion cell. And as we know, in this situation, because we are going to do with the colors, so cone cells will be involved, and they are saying that a single ganglion cell, this ganglion cell may be innervated by many cone cells like in the periphery, or may be innervated by few cone cells like in the central or the foveal region. So we know there are three types of cone cells. They are red cone cells, blue cone cells, and green cone cells. So they're saying that if the red, blue, and green, they are activated in all of them are activated in the same manner, like in exactly in equal proportions, what will happen? There will be a white signal generated. But what happens if only one color type of the cone is activated but another one is inhibited, due to which we'll have color perception? For example, there is a red and green cone excitation inhibition channel, in which red one causes excitation and green one causes inhibition. As a result, we detect certain type of color. And now for yellow and blue color, what we have is the excitation inhibition relationship between them is that blue cone is activated, whereas the red and green ones, there is inhibition of them. The red and green ones respond to yellow color, whereas the blue, it stands on its own. So they also make an excitation inhibition pair, and the red and green also make an excitation inhibition pair in another cycle. So the mechanism of this opposing or reciprocal inhibition or excitation inhibition, that one color causes excitation and other causes the negative signal, or it causes inhibition. How does this actually happen? They are saying that excitation will be a process which will be produced whenever there is direct signal transmission, and when there is direct signal transmission, it means from the cone cell the signal is directly sent to the bipolar cell. So this will be direct transmission, there will be excitation, or there will be depolarization in this mechanism. But in inhibition, what will happen? There will be indirect pathway that will be activated. The indirect pathway will cause the cone cells to reach the bipolar cells, but through horizontal cells, which will give an inhibitory output. As a result of this inhibitory output, there will be hyperpolarization. So that is how an excitation inhibition pair is formed in the cone cell pathway, and how we perceive different colors. So the importance of these color contrast mechanism allows the retina to start the differentiation of colors, and it makes the whole thing easier for the brain to detect the colors, and to identify or detect the colors. So that processing is done even before the color reaches the brain. That is, differential analysis is also done by the retina. Thus, color contrast type of ganglion cell is excited by one color, inhibited by another color, or the opponent color. Therefore, color analysis begins in the retina, and it is not entirely the function of a brain. So, this is how lateral inhibition, the black and white or achromatic vision, and the color analysis begins or is a function of the retina, rather wholly by the brain. With that, we come to the end of chapter number 51, the receptor and neural functions of the retina. I hope you understood each and everything related to different rod cells, cone cells, black and white vision, color vision, lateral inhibition, and the whole functions of the retina. I will meet you guys in the next video, that is with the chapter number 52, inshallah.